Hi, my name is Mr. Evangelista, and today we're going to be talking more about simple inheritance. So this is going to be simple inheritance part 2. Okay, so this sexy, good-looking guy right here is Gregor Mendel, and we talked about him last time, but he's considered the father of genetics, and this is why. So he did an experiment with pea plants, and he used pea plants because they reproduce quickly and they produce a lot of seeds. And so he took a tall pea plant and a short pea plant, and he noticed that their offspring were all tall pea plants, just like this one. This generation here we call the parental generation, and the offspring of the parental generation we call the F1 or the filial 1 generation. Now, he did something interesting. He took the F1 generation and bred those organisms together to create the F2 or the filial 2 generation. And he noticed that the short phenotype reappeared in the F2 generation. So 75% of the plants were the tall phenotype, but 25% of them came out with the short phenotype. Okay, so and then it's pretty interesting because Gregor Mendel did all this and he found all these two laws, which we're going to talk about now, but he knew nothing about DNA, about biochemistry, mitosis, meiosis, any of that, and yet he was able to figure out these two laws. And so here's a picture of meiosis, and in meiosis we start out with a diploid cell and then we end up with 4 haploid daughter cells. Now, if let's say the genotype of that original diploid cell was heterozygous, so big A, little a, and we know in meiosis we double our DNA, and then we figure out was these alleles separate from each other. And so here they are separated from each other. The importance of this is, well, when we're doing Punnett squares, we're not just doing Punnett squares just for the sake of, drawing a square. What it actually represents is kind of the randomness and the probability of meiosis and random fertilization. Meaning that, so these are going to be sperm, and if this sperm right here fertilizes this egg, then we get this genotype for our offspring. But it could be that this sperm would fertilize this egg, or if it was this sperm that fertilized this egg, or this sperm that fertilized this egg. That's why we're getting these probabilities. And so the law of segregation states that the alleles for each gene separate, and because these alleles have separated, we need to do a Punnett square, because since they're separated, they can combine and fertilize in random fashions, and that gives us the probabilities that we do in the Punnett square. Now, Mendel had a second law called the law of independent assortment, and it said that the different traits are inherited independently of each other. So what he wanted to figure out was, he had these seeds that were round and yellow, and he had these seeds that were green and wrinkled. And he was thinking, well, are seeds always going to be round and yellow, and are they always going to be wrinkled and green? Meaning, are the two traits linked to each other? And he found out through a cross here that they are not linked, and that the traits are independent of each other. Now, there are some traits that we see that are linked together, and we call them linked genes, such as if you have red hair, you're likely to have freckles. But these traits that Mendel were looking at were found to be independent of each other, and we call that the law of independent assortment. Okay, so let's take a look at a problem here. So we've already looked at simple genetics problems in the last video, and now we're going to look at this monster 4x4 Punnett square. This is called a dihybrid cross. Okay, so round is dominant to wrinkled, and yellow is dominant to green. What are the chances that a plant that is heterozygous for both traits, that is self-pollinated, produces a wrinkled green seed? By the way, first we start, what is self-pollination? So in plants, so if we look at this flower here, the anther has sperm. Well, it's not sperm, it's pollen. It's the same thing as sperm for plants. It's the male gamete, and we can get these pollen and we can actually put it inside the stigma. And inside the stigma here, it's interesting, we have eggs inside of the stigma, so you can actually self-pollinate a flower, get the pollen, the male gametes, and put them with the eggs, and pollinate the flower that way. And so here we have they're heterozygous for both traits, so big R, big Y, little y. And I'm going to show you how to do this using the big 16 Punnett square, and also how to do it using just the regular Punnett squares. So to use the big Punnett square, we have to think of all of the variations that we can get with these two traits. So we can get the big R with the big Y, and that's represented here. We can get the little r with the big Y, that's right here. We can get the big R with the small y, that's right here. And we can get the small r with the small y, and that's right here. And then, since they're both the same parents, they're both heterozygous, this parent is going to be the same as this parent. And from there, big R, big R, big R, big R, big Y, big Y, big Y, big Y. We fill out the Punnett squares that way. And if we notice, we're trying to find out what are the chances that we get a wrinkled green seed. So the only wrinkled green seed is right here. So that's 1 out of the 16 squares. So the chances are going to be 1 out of 16. Now, you can also do this by just doing the regular Punnett squares, and I'm going to show you how to do that now. So what we do is we just do the Punnett squares for each of these traits individually. So they're both heterozygous. So we're going to cross a heterozygous round with a heterozygous round. And if we do the, look at that awesome box. If we do the Punnett square for this, it's going to be big R, big R. This is going to be heterozygous, heterozygous, and homozygous recessive here. So what are the chances we get a small r wrinkled seed? Well, it's 1 in 4 here. So 1/4 chance to get a wrinkled seed. Now we're going to do the Punnett square for the color. And again, the trait is heterozygous, so it's big Y, little y, big Y, little y, because we're self-pollinating. And so let's do this. We have big Y, homozygous dominant. We have the heterozygous, and homozygous recessive here. Okay, so what are the chances we get a green seed? Well, the only way we get a green seed is if we get the homozygous recessive. So here is the green seed, and that's going to be 1/4. Okay, and since we want wrinkled and green, whenever you hear and, that's going to mean you're going to multiply. So here, if I multiply 1 times 1 is 1, 4 times 4 is 16. And we get the same answer, 1 in 16 chance to get that wrinkled green seed. Now the last thing we're going to talk about is how to do a test cross. Now a test cross is important if there is an organism and you're trying to figure out what its genotype is. Now if the phenotype of the individual has the recessive phenotype, it's easy because the only way you get the recessive phenotype is if you have the homozygous recessive genotype. So if it has the recessive phenotype, you're going to know it's going to be little p, little p. But what if it was the dominant phenotype? Well, there are two options if it's the dominant phenotype. It could be big P, or it could be heterozygous big P, little p. And how do you figure out which one it is? Well, the way you do that is you do a test cross, meaning you cross it with a homozygous recessive, that was a horrible line, homozygous recessive individual. And when you do that, this is what you get. Well, if it were big P, if the genotype of the organism were big P, then all of the offspring would come out purple. They would all come out heterozygous here. And so if you came out with all heterozygous, well, then you know that the original genotype of the parent was big P, homozygous dominant. If half of them come out the dominant phenotype and half of them come out with the recessive phenotype, well then you know that the original genotype of the parent was heterozygous, big P, little p. That's it for today, and I hope that helps.